typical question that I was getting in talks, and not just me, but all people in my lab, was, have you corrected for gender? And you can correct for a lot of things, and you can start correcting everything out of everything, and you can be left with no variance at all if you start pretty much correcting for everything that you can find. But the key question is, if there is something important, this is a variable that we care about, why correct for it? We can actually understand how it affects expression. So what we do is, instead of just taking a population that is like this, so we don't actually see the gender, we actually split it in males and females. And what we do is we assess the discovery of EQTLs in its individual set relative to what we discover in the total population. So we're looking for gender-specific activity of EQTLs in the same way that we look for tissue-specific activity. We also find evidence for epistasis. And when I say epistasis, I mean the modulation of a non-synonymous SNP by an EQTL SNP. Basically, we published before that we expect about 18.2 non-synonymous SNPs to be modulated. By assessing allele-specific expression, we find that 23.3% of non-synonymous SNPs are actually modulated with differential allelic expression. Now, why is this important? Well, this is important because when we go and embark into all these studies that look for rare variants, and we're going to be looking at non-synonymous variants, essentially we'll be taking each variant as it is. We'll be counting 1, 2, 3, 4, 5. But it is important how much the specific haplotype that carries the variant is expressed. So if, for example, that rare variant is expressed higher than the relative normal variant, should be weighted differently than if it's the other way around, or if there's absolutely no differential expression. And it's the same thing as if you think about a monogenic disorder effect. For example, if this is a disease variant for a recessive disorder, pretty much this acts like a homozygote because the vast majority of the protein is actually coming out of that disease haplotype. So there are ways actually that we can improve the information, especially if we have the relevant issue for a given disease, to weigh differently those particular effects with rare variants. So in the last few minutes, I want to talk about a new direction that we're taking, which is to look for rare EQTLs. Now, of course, EQTLs to discover with common variants has been quite a trivial exercise now. We have large samples, we're able to assay different tissues, and we can do the association in a straightforward way and discover those rare variants, those EQTL variants, and we can actually get probably to the causal variant once we have full sequencing data, or now that we have full sequencing data.
In the last years, the number of genome-wide analyses has greatly increased thanks to the new technologies and generating a huge amount of data. But most of these studies have focused on coding regions that represent only the 5% of the genome. So there is a lot of studies that analyze the rest of the genome, especially repetitive elements that constitute the 50% of the genome. The most abundant family of repetitive elements is ALU families. ALUs are retotransposons that are specifics of primates. There are more than 1 million of ALUs per haploid genome, that is the 10% of the genome. They map to rich genes regions. They contain the 30% of all CPG sites, and most of them are methylated. Depending on where the ALU is integrated, they can affect the expression of the gene. They can add, for example, new regulatory elements when they are in the promoters, or if they are in the gene body, they can affect the alternative splicing, the RNA editing, they can truncate the expression of the gene, etc. We can say then that there are 5,000, at least 5,000 fully unmethylated ALUs that are potential genomic regulators or disruptors in normal cells, and up to 20,000 in cancer cells. Our preliminary results suggest that unmethylated ALUs constitute a defined compartment within alum with specific features. Here, these are results that I have not presented, but we have performed this analysis, and we have seen that unmethylated ALUs have a higher degree of conservation than methylated ALUs. They also have a low sequence divergence and a high GC content and CPG observed versus expected index. They are located near CPG island or transcription star sites, and they lie in active chromatin domains enriched in trimethylation of lysine 4 in histone 3 and acetylation of lysine 9 in histone 3. What we did was
essentially came out with a very simple filtering scheme. So while we're looking at things on a variant basis, what we would like to do is group them by gene. So if we just look at any one person, we have maybe 20,000 genes. We can then narrow that down to genes which have a protein coding variant. So this actually comes down to maybe about 4,000, which is still quite a lot. So what we then did was just filter down even more to look for genes with a protein coding variant that we think is novel. And this comes down to maybe about 400. And what we can then do is intersect these the smaller set among different individuals, assuming that the same gene will be mutated among individuals. So there are a number of assumptions that we're making right here. First off, that that disease gene is, that our disease mutation would be coding. So that's a major assumption that we're making. The second one is that it's highly penetrant, such that we're not going to see it in any databases or any other controls. So this is a table showing what I showed in a graphic earlier. The first thing we're asking is what are the number of genes which we have at least one protein altering variant, and we define that as non-synonymous, coding indel or splice site variant. And then right at the bottom, we require that the variant is not in dbSNP, or at that time, not in 8 HapMap control exomes. And what we chose to do was Miller syndrome, also known as post-axial acrofacial dysostosis syndrome, first described in Seattle, 1979. And I think Steve from Complete Genomics mentioned that in parallel to the exome sequencing that we were doing, they were also doing whole genome. So just some brief facts about Miller syndrome. The main phenotype that we're looking at were the limb defects in the syndrome, and based on the background that we knew, it was very likely to be autosomal recessive. The interesting thing about that was that most of the families that were described were non-consanguineous. Actually, all of them were non-consanguineous. The closest related parents in any of the described individuals were fifth cousins. And this is very unusual for what we think is a recessive disorder.
Okay. I will start with the conclusions of my talk. Well, the first conclusion is very clear from the previous talk, so I'm not giving conclusions about Sarah's work, but actually also about our work. And that is that exome sequencing indeed is an ideal tool for Mendelian disease gene identification studies. Of course, it will miss everything outside of genes. I think the most important conclusion for myself from the last year is actually that de novo mutations are a very frequent cause of mental retardation, and I hope to show that to you in this talk. I think that's the most important novel finding. And as the title of my talk said, we are really dedicated to making this into a clinical study, and I think exome sequencing really has a very high potential for becoming a very routine approach in diagnostics, and I think it's better suited than, for example, more targeted approaches. And the last conclusion is that actually we heard quite some big science talks in the last few days, and I hope to show you that actually this work can be done outside of genome centers in individual departments.
This is the work of a former postdoc of mine, Michel Samet, who is now a group leader at the New Sequencing Center in Barcelona. Let's assume that this is a gene, and these are all the different alternative transcript forms of the gene. What we do, we create, it's called a splicing graph or a Lux network or whatever. We use different names in which each edge corresponds to an exon or to an intron junction, right? And then what we do, right, we just simply map the reads into this network. So here we have all the reads that map into this exon. And here we have all the reads that map across this intron junction. And then we do, as I just said, we just assume that the number of reads that maps to this exon is proportional to the sum of abundances of the transcripts that share this exon. So for instance, the number of reads that I see mapping here, right, is the sum of the abundance of this transcript, and this transcript. Plus, actually, some error, because the problem is not that the system is, the problem usually is not that the system is underdetermined, it's actually that it's overdetermined, that it's inconsistent, that it's really impossible to find a solution that satisfies all the equations unless you use some additional error, right, to make the equations work. So at each of the nodes, at each of the edges of this graph, we just have an equation, right? An equation that has all these unknowns, 1, 2, 3, 4, 5, 6, and then we using the standard systems, we solve the system of linear equations, and then we attach abundances to each of these transcripts. We have been working actually quite a lot on this set of genes. Actually, this is mostly the work of Thomas de Rien in my group as a postdoc that's now back in France, and now the work is being followed by Andrea Tanzer, a postdoc in my group as well. And I mean, we started by selecting, this was an early release of this annotation. It had only a few long non-coding RNAs, about 5,000, and now, as I said, we have 10,000. And we use only the ones that were intergenic, the more equivalent one to the set of link RNAs, and we end up with a set of about 3,000 long non-coding RNAs, which I think I want to emphasize the importance of this data set. These are long non-coding RNAs for which we know the sequence